Today I want to give you my view on root traits in crop breeding, where we are and where we would like to be. Root traits in breeding or root traits in general have gained considerable attention in the last 15 years or so, and was even postulated that root and root traits would be part of the second grain revolution that would produce crops that are tolerant to stresses and nutrient efficient and so forth. And so there's been quite a bit of research, and there has been quite a bit of investment into read phenotyping platforms such as these. And as part of the thinking process, root idiotypes have been put forward. So idiotypes of root system architectures that are adapted to certain abiotic stresses. So for example, under drought stress, we would have plants with deeper roots in the nitrogen deficiency, or to capture nitrate in soil. Deeper roots would also be beneficial, whereas under phosphorus deficiency, shallower roots would be beneficial because P is more concentrated in the topsoil layer. Now, these have also been partly proven experimentally. The problem is, of course, that in agriculture, in the field, the system is not static. So if we have a well-watered field, yes, a shallow root system would be capable of taking up more P. A deeper would be taking up more water and nitrate. But what happens if we have a drought, such as here? All these shallow roots all of a sudden find themselves in a medium where there's insufficient water even for phosphorus diffusion. So the whole concept will break down if we have changing environments and multiple stresses at the same time. And this has plagued this area of root research significantly. There are other interactions between traits where we clearly see this trade-off. For example, narrow xylem vessels have been put forward as a way to improve drought tolerance, but on the other hand, thicker primary roots with a bigger central cylinder are considered key for improving yield potential. So clearly a trade-off there. So in a constantly changing environment in the field, these single extreme traits may not be the solution. And instead, we may have to consider more dynamic traits. And among these dynamic traits, there's been the concept of root plasticity, so a root system that is adapting to a changing environment, but is experimentally very difficult, as you can imagine. And so there is very little progress made there. We may then think of traits that specifically address environmental changes. For example, there has been quite a bit of research on drought recovery. So what are the traits and what is the speed at which a crop is recovering to full functionality after a drought period? And my own team, we have looked, for example, as a number and the speed of new root development following a drought. And then there are other traits like the low-cost root system here, potentially due to development of parenchyma, which would be a beneficial trait possibly under many different environmental scenarios. And of course, if you look at, or if you want to consider root traits in breeding, we need to consider the breeding process per se. And we can divide it into basic 3 phases. In the phase 1, there's at the start of any breeding process, there's a cross between 2 parents, most likely. And one parent is typically a high-yielding adapted variety to the target region, and the second parent might be a donor of a new trait of interest, for a root trait, for example. So here the choice of the donor would be the interesting question in our context. And then this is followed by a second phase, in which plants start as genetically highly heterozygous, and in every generation they become more homozygous and more stable. And this period is, because of this heterozygosity, it is still a bit difficult to do reliable selection, but this period is extremely important in the breeding process because it's the period of recombination, where genes from parent 1 and parent 2 are reshuffled in novel combinations. So we need to go through this period to allow these new recombinations, and selection here is predominantly there to fix useful genes and discard really unacceptable types. And then this is followed by the third period, where lines are homozygous, where we can have uniform larger plot sizes and where we can select for yield in the end. And you can see, so the selection process typically goes until the F8, I would say, plus minus 1 generation. And then after that, there's multi-location testing. So everything takes at least 10 generations, or 12, let's say. And we can ask different research questions for each of these phases. So here in the first phase, questions would be regarding the donor identification. And the first most important question probably is, what are the root traits that are most likely going to improve our field performance? Which are the root traits that we should read for or at least select for in a donor? If we have decided what the traits are, the next question is, does enough variation exist for these traits so that we can identify different or donors with a high impression of these traits? And then the question is about phenotyping. Of course, the precision and the throughput, which is also the most important question if you would ever consider phenotyping for root traits in the second phase here, the throughput and the precision question. To sort of circumvent this problem, one option is to replace phenotypic selection by marker-assisted selection. So if the trait is controlled by a few major genes, we can develop markers that are diagnostic for these genes and select for these rather than selecting for the phenotype in the field. And this concept can be brought a little bit further, even for polygenic traits, by a concept of genomic selection. I'll briefly talk about that. And then the research questions here, when we have already somewhat larger plots, we can ask, can we really phenotype root traits either directly or more likely indirectly? Indirect would be, for example, by imaging of canopy, temperature, or other indirect traits. Now, I want to go through some of these different processes at these different stages and want to begin with the question of what are the traits that we might be interested in, and then come to the point of the donor selection and how that would work in breeding in the end. I already mentioned that a low-cost root would be a potential useful trait. So we have addressed that in our own work with rice. Rice has a quite complex root system for cereal. It has the main roots, like this is the primary root. It has crown roots. And then from these crown roots, we have two types of lateral roots. And that's the special thing for rice. It has the larger L-type roots that may branch again into other L-types. But then it has a second type of lateral roots. They're very fine, small S-type laterals that branch off every other root type. And then if we zoom in a little bit more, we need a microscope to do so, we see that there's hairs growing on crown roots. This is an L-type, this is an S-type. So even the smallest S-types still branch further into root hairs. And for a nutrient like phosphorus uptake, the exploration of the soil volume through these fine structures is potentially very important. And here you can see that there's a clear decrease in length, but and in density in these root classes. Okay, now we have developed a model, a 3D or 4D model of a rice root that we can then use to model phosphorus uptake processes in the soil, and we can ask questions about costs and efficiency of different root types. So we have modeled the uptake of hairs of this very fine lateral roots, the large lateral roots, crown roots, and we see that crown roots take up the most P, and it's simply because they have the largest surface area, and phosphorus uptake is always proportional to the surface area. But because they are big, they also incur a large cost in terms of assimilates. And if we work particularly on P deficiency, where P is limiting plant growth, then P is a cost that the plant has to consider in terms of where it is investing the P. Because a crown root, for example, would incur a much, much larger cost in terms of the P needed to build the root than in a fine root. So from that, we can model the benefit-cost ratio, and we can see that the fine structures are, of course, highly beneficial. And we can also look at this from a slightly different angle, the cost recovery time in days. So the P invested to make hairs and these very fine S-type roots will be recovered through the P uptake of these fine structures within a day or two. Whereas a crown root, the cost to make a crown root is only recovered by the P uptake of that crown root after a month or so. So we clearly see that if we want to have a root system that is capable of responding to P deficiency, the most efficient way would be to do it to respond by producing low-cost structures rather than the high-cost structures. And of course, overall, there needs to be a balance between the two. But based on this modeling, we have the hypothesis that these fine root structures are beneficial. And then we have a second model that is more complicated in the terms of the soil process that are taken into account in the phosphorus uptake process. It includes is solubilization process of P in the soil. So P is typically tightly bound in the soil, and if the plant can solubilize and release some of this P, this will be very beneficial and important for phosphorus uptake. And the solubilization occurs as a result of a pH increase driven by excess nitrate over ammonium uptake. And so the excess negative charge taken up is balanced by a negative charge released from the root, most likely bicarbonate. And this bicarbonate will then increase the pH in the rhizosphere, and that will release phosphorus, and that phosphorus becomes available for plant uptake. The catch now is that the pH charge will diffuse quite quickly out from the root, whereas the diffusion of P is very, very limited in the soil because of this tight binding. So we can assume that the P solubilized in close proximity to the main root diffuses back, but the P solubilized here by the pH change will take too long to diffuse back to the crown root and will not be taken up immediately. And now here the fine root structures come in. If a plant has a dense cover of S-types, and if these S-types have a lot of root hairs, these will be able to mop up the P solubilized by the parent root. So again, a potential important role of these fine root structures. So based on trying to understand important traits, through modeling, we can see that lateral roots and the root hairs contribute most to phosphorus uptake. And they do so with little cost in terms of P invested, and should therefore be traits that breeders should consider. And now the first question that is really related more to the breeding part is, does genetic or genotypic variation for these traits exist? And we've looked at that. We've looked at root hair length, for example, and yes, there is a bit of variation for this trait, but there is even more variation within a genotype, and clearly this is not a trait that is suitable for breeding, I think. Whereas the variation in the lateral root density does seem far more promising. And you can see here this is variation for the S-types. And then here these thicker ones are the L-type lateral roots. And for both we have clear variation in between these genotypes. And we have made a cross between this low density Nerica 4 with this high density NDG 188 and developed the QTL mapping population and looked for genes and markers in this QTL mapping population. And here are the results of this study. It was a QTL mapping study with about 120 lines. They were grown in the field. We've dug them out from the field, from a P-deficient field. Roots were scanned and then analyzed with image data. This was certainly not a very high-tech, high-throughput process. But in the end, I think it was worth the effort. We identified several QTLs, in particular the one that are highlighted here in green for L-type density on crown roots. This had a major effect. It explains more than 40% of the variation for the trait and is statistically highly significant. Before we can use this QTL and the linked markers in breeding, we still need to go through a process of confirmation of the QTL. to convince ourselves and to convince the breeder that it is really worth investing into marker-assisted selection based on this QTL. So there needs to be a genetic confirmation, and that is, I will not talk about that now, but in terms of the physiological or the phenotypic confirmation, I can show you some data. For example, as I said, we have looked at many different root traits in our mapping population. And through some multiple regression, we can ask which of these root traits really contribute most to phosphorus uptake. So this was a multiple regression of root traits against phosphorus uptake. And we see that the L-type density on crown roots was the most influential trait, followed by crown root number and the S-type density. And a cumulative model explains almost 70% of the variation in phosphorus uptake in the mapping population. And we have then developed a simple root index based on the variation for these three traits. And again, we see we have an R-square of 0.7. So we can, with relatively good precision, predict the phosphorus uptake of a breeding line based on these three traits. So now we could, in theory, go ahead and move to marker-assisted selection, at least for these two main QTL that we identified. Now you see the study was done or was published this year, so it's all relatively recent results. And I cannot show you any progress in the marker-assisted selection, but to give you an idea how marker-assisted selection would work, I go to an older story of mine where we have actually already completed the whole process. And PAP1 was a locus or QTL identified a long time ago by me. And it stands for phosphorus uptake. And it's a QTL derived in a population of cassalas as a donor. Cassalas is a traditional rice variety that no farmer would really grow anymore, but it's one of these gene bank accessions that harbors potentially useful genes. And it was crossed with a modern variety that has high yield potential when it's well fertilized, but on this soil, which is very high P fixing and it's not fertilized, you can see this doesn't produce even any yield. So we have identified this QTL, and then through several years of genetic studies, we identified the underlying gene. It's a kinase that we termed pistil-1. We understand that pistil-1 is expressed at the very earliest stages of crown root and other root development. And you can see the expression here in these crown root primordia. And high expression of this gene helps to maintain root growth under P deficiency. And if a plant is able to maintain root growth, it's also able to maintain phosphorus uptake. And here you can see there's IR64. It's a famous rice variety that lacks this PAP1. And if you transform it, so this is a transgenic plant, it grows much, much better. But since it is a natural occurring allele, a natural occurring variant, we can also achieve the same thing through conventional breeding and marker-assisted selection. So this is what we have done. We have crossed IR64 with the donor of PAP1, and then we use these markers to make our selection. And since the donor, this cassalas, is not a very desirable genotype, we do several back crosses to reproduce the more desirable IR64 to some extent at least. And in each of these generations, we use the markers to make the selection. And at the end of this backcross process, we have different IR64 lines that all have PAP1 already fixed. But they still vary for other traits like plant height and maturity and this and that. And here you can see this is the intermediate result. This is IR64 in a low P field, and this is the PAP1 line in the same low P field growing much better. And now we've crossed and we've selected two of these PAP1 lines, we've crossed them, and we sent the seeds to Madagascar, where we then continued conventional selection for those to benefit from the remaining variation that is in this population. And we've done selection under really quite harsh environments without fertilizer in very P deficient fields. We involved farmers in our selection for plant type and panicle type, and we even involved them in tests of taste and appearance of the rice grain. And as a result of this marker-assisted selection, combined with selection in the target environment, we were able to release a new variety in Madagascar. It's called Fivari 32. It was released 2 years ago. It's highly tolerant to P deficiency because of the genetic background of IR64, it's also very responsive to P fertilizer, and it is now spreading in the country. It's promoted by the Ministry of Agriculture, and just last season we produced 40 tons of seed to be distributed to farmers there locally. So a nice success story of how marker-assisted selection can be used even for complicated root traits. Now, I want to show a somewhat different example where markers are not involved because we don't have strong QTLs. We don't understand the trait enough to have markers for the selection. But in this case, we just focus on the donor. And our donor is DG123. Again, it's a traditional variety from the gene bank. And it's a donor for multiple traits. It has high internal P utilization efficiency, so it makes more biomass per unit P taken up, which basically relates to low-cost roots and low-cost shoots. It has higher P acquisition efficiency, so per unit root size, it takes up more P than another variety, Nerica 4, for example. It has better root branching compared to Nerica 4. It has slightly longer root hairs, and it has a different root angle distribution. So these are all traits that make it a potential useful donor in our breeding, and we've developed several of these populations. For example, we've crossed DJ with IR64 again as targeting the lowland rice. We've crossed DJ with Nerica 4. Nerica 4 is a commonly used upland variety in Africa, but that lacks adaptation to P deficiency. So we've made these crosses. We've sent the F3 seeds to Madagascar again, but because we don't have markers, all we can do is conventional selection, but importantly in the target environment. So in low P fields, in farmers' fields without fertilizer, under their management. And this is a lengthy process. Again, I mean, from the F3, it takes about the F8 of conventional selection and then multi-location testing. But also this approach, this sort of blind black box approach works. And we've released one line from this cross as Fivari 85, and it's already available to farmers in Madagascar. And we've also developed an upland variety that has just been registered as Mavitrica in Madagascar a few months ago. And because it is, in this whole process, we really just select for appearance and for yield. We don't look at root traits. What we can do then in the end, finally, we can see what did our selection do to root traits. And here we can, so for the more or less finished product, we can have a look. And we see that Mavitrica has, for example, much faster root elongation than Nerica 4. And more importantly, for the phosphorus uptake, it has more efficient roots. So the roots, this is the blue is Nerica 4, the green is the donor DJ, and the orange line is Mavitrica. So it is more efficient in taking up P per root biomass or per root length than the others. And so this is something we achieved unknowingly through our selection. And then I already mentioned that DJ has a different root distribution, and we think this is potentially interesting for a whole different topic. We've crossed it to X265, which is the main recommended rice variety for Madagascar. And X265 is one of these typical lowland varieties with a very shallow root system. So we've tested the angle distribution by burying these sieves in the soil. We plant a seedling into this sieve, and then after 40, 50 days, we can dig up the sieves, and the root distribution is held in place, and we can study the different angle distributions. If we remove all the shallow angles in X265, there's just a few roots left that are growing almost vertically. And of course, in DJ, you see there is a much higher distribution of these vertical roots. But still a very even distribution of roots everywhere. And we think this might be an important trait for adaptation to droughts, to water shortages. And we are now doing selection in this population under this kind of conditions, where rather than transplanting rice into a flooded field, we do the direct sowing into a field that may experience drought at any time during seedling development. And this again could be a way of using a donor of root traits in a sort of blind box selection approach. So far, mostly the phenotyping for root traits was done in detail for a few parents and few parents to identify donors. And we have not yet really done any smart phenotyping in larger panels, and we have not provided any solution for selection for root traits, at least. So while joining the PhenoRob team, we also had an offer to work with a rice diversity panel and with some images that were provided by friends of mine at Rothamsted. So our resource that my PhD student Lukas Grusenbaum is working on, the resource is 600 lines of this rice diversity panel that is completely sequenced. So we have a lot of markers available for this panel, suitable for any type of genomic analysis. And we have these seedling root images of 600 of these accessions. And after some quality filtering, at least we still have 346 genotypes left, 870 images to analyze. And our intention was to do machine learning for this phenotyping. And while to the human eye, it is very clear that there is a big difference in the length of lateral roots and the density of lateral roots between these genotypes or between these genotypes. Actually, the machine learning approach failed us in this regard. And the problem is that we can phenotype this one with relatively good precision, but whenever things become too dense and hairs become too long and everything starts to overlap, the machine learning approach or any fast approach failed in these denser types that are actually desired types. So instead of following the machine learning approach, we went back to more phenotyping by hand on these images, and Lukas came up with several root traits that he measured on these. And the only type of optimization was through some custom macros for image J, but it still was a lot of phenotyping by hand in the end. And we came up with a longer list of traits from root length to S-type density, L-type density, and so forth. And now I want to introduce you to a different way of getting at understanding the genetic control of potential traits and using this understanding in selection. And that is genomic prediction. So in QTL mapping, we try to find one marker that is linked to one particular gene, or a few markers linked to a few genes. Genomic prediction tries to cover the whole genome without really giving particular emphasis on any strong genes. And so how it works is we have a genotyping or a genetic data sets with genotyping data, in this case from 3,000 sequenced accessions of the Erie gene bank. It's the biggest rice gene bank in the world. And then we phenotype a subset of that, in this case, a little bit more than 10% of all the available accessions. And this is our training population. And from these two data sets combined, we develop a genomic prediction model that, based on the genotype data, then predicts the expected phenotypes for our root traits. And in the first test, we verify that in a test set. And then in the second step, and this is really the final outcome that we want to achieve, we try to predict the root phenotypes in the 90% of the population that we didn't test phenotypically. So it's a very efficient way of looking at a large number of lines by only phenotyping 10% or so. The same, in principle, the same process can also be used in the selection process in breeding as well, but we are not yet there, so I will not talk about that. But I will talk about the verification and the prediction now. So in the first step, we look at the accuracy in the test set of our prediction, and typically for some of the important traits, we are at an overall accuracy of about 0.5. So that would correspond to an R-square of 0.5, where here we have the measured values and these are the predicted values. And unfortunately, this is a bit of an overestimation, and I don't want to get into detail of that. We still have to work on optimize some processes here. But what we wanted to do is we wanted to predict root traits in this whole set of 3,000 or the remaining 2,660 in a search for new donors. So that is what we can do. Here is the distribution of the 2,600 or 3,000 for lateral root density, the L-type density is for the S-type density. And we can now select the top 1%, for example, for each of these traits, order them from the IRI gene bank, send them to Madagascar, send them to Bonn for verification experiments in the target environment. And this is what we're actually currently doing. And in March, Lucas and I will go to Madagascar to do the field work there. And then next spring, we'll do some more detailed studies here in Bonn in the greenhouse. Okay, so let me come to the first part of my conclusions about where we currently are. And you have probably noticed that most efforts that I've talked about, and I think most efforts that are currently ongoing in terms of root traits in breeding, are directed towards the identification of useful traits and donors for these traits and possibly marker identification. And all of these activities actually have nothing to do with the selection process per se. They are all prior to the identification of the donor. So we call this pre-breeding. So really, unfortunately, all these activities are currently focusing on the pre-breeding activities, where we hope to identify also QTLs and GWAS peaks in order to identify loci for marker-assisted selection, so that at least we can move into this realm here and use the markers that we identified in pre-breeding. The problem why we possibly have not been able to use any selection, direct selection in the breeding process is also simply the problem of size. And that's due to the recurring nature of selection breeding. Every year, new F2s are produced and need to be funneled through the system. So every year we have to go through the system, whereas once we have identified donors and markers, this is a one-time effort. So much more efficient in terms of resources than phenotyping these every year. And there's not only one population, but a breeder typically works on a few dozen different populations in parallel. So this would have to be multiplied by 10 or 20. So clearly a limitation to breeding. So where we would like to be is to actually move a little bit more in the phenotyping towards this part of the breeding process. And the question, of course, is the question of throughput and of heritability and the precision of the phenotyping. And so far, I think what is working and what is employed in breeding is some very indirect way of looking at root traits. For example, for using canopy temperature or some spectral imaging to make very educated guesses about underlying root traits. So here a cool temperature would suggest that water uptake is proceeding, and that probably would suggest that there's a better root distribution of depth in this particular genotype. And the ground penetrating radar that is colleagues are working here within the FINRO project could possibly achieve something similar. But still, these would not be really direct selection for root traits in the field, but really rather indirect selection. So where do I see the direct selection or direct phenotyping of root traits to occur? I think what we might achieve is more precision phenotyping of root traits in the pre-breeding process. So to be better able to select donors for the subsequent breeding process. And what we need to achieve is associate root traits with field performance directly and really come to the point to say that this root trait will improve yield by this %. And to convey this message to a breeder so that he actually starts to use the donors and whatever methodology we can provide him in the actual breeding process. A lot of effort, I mean, this has been, of course, recognized, and quite a bit of effort has been devoted to look at 3D or even 4D root distribution using very fancy methodology like MRI or CT scans, for example, and that have produced images like these where we can actually see the root distribution in the soil. The problem that I have a little bit with this approach is that when you look at this rather vertical root distribution and you compare it with the distribution that we actually observe in a breeding field or in a field where we have just washed off the top 10 centimeters of soil, you can see there's a very clear difference in the distribution. If these plants wouldn't have been grown in a container that clearly limits the lateral root extension, we would have all of these roots that bend down here, they would have grown horizontally. So this 3D root distribution estimates obtained by these methods are clearly limited by the container and are probably only reliable at the very early seedling stage. And I hope that maybe with using the SEIT method developed by our colleagues in geoscience, maybe this will offer a solution in the future to actually look at this distribution in the soil in a more realistic way. So this brings me to my last slide, to my final conclusion. I think we probably all agree that improving root rates will be key to adapting crops to a water nutrient-limited future. But the question is how to achieve this considering the limitations in the breeding process, which is basically we have to do the breeding process to some extent in the field, and we have a very large number of lines to look at. Where we need to make progress is really identifying most influential traits. Influential traits for yield, not only for an intermediate process, but really for yield, that are influential across years with changing weather patterns. So really traits that a breeder would take seriously. Then, of course, we need to look at the variation and select donors for that, and that might be more of a technical problem in terms of phenotyping. Ideally, we would identify markers to still avoid the very costly phenotyping in the field, and we would need to require medium throughput, efficient throughput of 150 to 300 lines times some replicates, so the number will increase. Alternatively, I think we can make progress with this genomic selection in the future for traits that are controlled not by few but many genes. And the question really is, will we ever be able to directly select for roots in the breeding process, in the selection process? And this will be clearly a question of cost, not yet available technology, and the question of heritability. And if you ask me what is my take on the most serious bottleneck, I think it is this one here. We need to identify the traits that really contribute to yield in the field. Once we have identified these traits, the rest are technical problems that we probably can solve. And with that, I'm at the end of my talk, and I thank you for your attention and happy to answer any questions that you may have.